Okay, so I apologize if this is a little bit difficult to hear. There's a lot of background noise, so I do apologize. And so before we actually use microscopes in class, which will be soon, I want to go through the parts and how it's used. And this will also be like on your quiz next week. I think our quiz is on block day next week. So I'm likely to be scribbling, so I do apologize for that. All right, so the microscope that we use in class is called a light compound light microscope, and that means that we're using the compound part refers to the fact that we're going to be using multiple lenses to put light onto our specimen to magnify it. And a light microscope can magnify about up to several hundred times. So, in the case for these microscopes, we're going to go anywhere from about 40 to about 400 times magnification for the microscopes that we have. So, it has a pretty decent magnification power. And we need to make sure that our specimens are very thin. So, like, you can't just stick like a chunk of liver up there, or even like if you think of onions in layers, you can't take like a whole layer of onion that's too thick. You need just like the skin off the onion. So it really has to be just a one or couple cell layers thick in order for you to have that light pass up through it. Um, so you need to make sure that the specimens are very thin so the light can pass through it. All right. And then we'll talk about resolution on another slide. And we'll spend a separate time talking about electron microscopes versus the compound light microscopes. I'm not going to talk too much or try not to talk too much about electron microscopes at this point. All right. So we're going to first start with the parts. And while it gives us a word list here, there's some that I'm going to switch around and there's some that I'm going to add to it. So um, just keep that in mind. So the first part that we're going to go through, and I wish this had like a text I can type, but I don't think that it does. No, I have to scribble it out here. Again, I wish I had my stylus, but all right. So this lens here, it's not actually going to be called an eyepiece lens. I mean, I think if you said that, I would know what you were talking about, but we're going to use kind of the more scientific term here and call this an ocular lens. So an ocular lens um, is the part that your eye is going to look through. And ocular lenses usually have a 10 times magnification, but this one is that we have might be a little bit different in class. So I'm just going to write down approximately 10 times magnification. That's going to be important in a little bit. But the ones we have are some of them are 20, some of them are 10, some of them are like 14. So they can vary. All right. And so that's going to be the lens that your eye looks through. Um, let's actually, I'm not going to go in. Yeah, let me go just in the order that they have prescribed here. This next part is called the arm. And I also want you guys to know like what these things do. So please just listen for what it's doing and jot these notes on underneath here somewhere. I just can't write all of that out at once on this particular video. So the arm, and I'll just cross that off as we get to it. The arm is going to be a place where you use your hand to hold on to it. So you're going to put one, we're always going to use two hands on the microscope. And you're going to use one on the arm and one on the base. So you're going to hold it. Imagine that this is my microscope and we are going to hold one hand on the arm and one hand on the base. Okay. Like that. Hold it close to your body. Don't be having it tilt. We don't want it to break. All right. Next structure here on the microscope, the next part, this is called the coarse adjustment knob. So it's COARSE, coarse adjustment knob. And the course adjustment knob is going to be what you're going to use to focus. Sometimes it's called a course focus knob. There's a course focus knob, course adjustment. That's what I prefer to use. And that is going to be used to focus your specimen when you are on all but high powers. So I'm going to do this and say that we're going to use course adjustment when we are on 10x, and I'll explain that in a little bit. When we are, which is the red objective. No, sorry, I messed that up. Let me see if I can get an eraser here. So when we are on 4x, when we are on 10x, those are the only two objectives, the red and the yellow, that we're going to use the course adjustment knob for. So we are never, ever, ever going to use that course adjustment knob when we are on the blue objective. So when we are on the blue objective, we can use what's called the fine adjustment. So I will write this in blue so you can see it, that this is the objective that is going to be the 40x. So you never ever use the course adjustment when you're on high power or 40x. You should only use the fine adjustment when you are on the 40x or high power objective. Now, adjustment. Okay. So that is my fine focus or fine adjustment knob. And you can use the fine adjustment on the other ones. It's just that you can't use the course on when you're on the you can't use the course adjustment when you're under high power. So, and really, it's not going to help that much to use the fine adjustment when you're on the lower powers anyway. So I tend to use the course adjustment exclusively when I'm on 4x and 10x, and only use you can only use fine adjustment when you're under high power. All right, so when I'm talking about high power, this 410 and 40x, what I am doing there, or what I'm talking about is, and I'm not switching colors for any particular reason here, we have these lenses here, and there's usually 3 or 4 of them. It depends on what microscope we have. The ones we have in our class are going to have, there's only 3 on them. So the 4x, the 10x, and the 40x. But what you have to realize is that to get the total magnification, and we'll go through some total magnification powers in a little bit, but to get the total magnification, you have to multiply these are called objective lenses. Objective lenses. You have to multiply these by the ocular lens. So our objective lenses have anywhere from 4, 10, and 40 times magnification, but you have to multiply that by the ocular lens. So to get the total magnification, you multiply objective times the ocular. So if your ocular is 10, then it's going to be 40 total, 100 total, 400 total times magnification. So these are on, actually, I want to add a piece to this that you should know. It's also be on a quiz. This is something called a revolving nose piece. And so that, as its name implies, it is going to revolve to turn the microscope objective lenses into place. Revolving nose piece. All right. Now, this bar here that they're pointing to is something called a mechanical stage, which our microscopes don't have. Ours do have a stage with some stage clips. It's not as fancy as this one, but that's okay. It gets the job done. So this is called, this part, the whole part that the slide is going to rest on is called the stage. Just kind of like a little platform that the specimen will be on, so the light can pass through and the lenses can look through the thin specimen. So the stage is where we're going to place our slides. All right. And then So that is the stage. Don't worry about mechanical stage part of it. We got objective lens here. And I'm going to cross this off totally, condenser. It's not, our microscope doesn't have one of those. But underneath the stage, it has a circle that has different size holes in the circle. So it regulates the amount of light that passes through to get to the stage. So sometimes this adjustment, this is called an iris diaphragm, and sometimes it's like a dimmer switch that can regulate the amount of light that reaches the specimen. But ours is going to be, like I said, these little circle that has different size holes in the circle that regulates the amount of light that goes through. So some of these are going to, some microscopes have it where it just continuously closes. So this is going to let less light in, and then this is going to let more light in. This one that we have, you have to rotate it. So different size holes are going to be lined up with the hole that's on the top of the stage. They're just two different ways of doing it. So, all right, so that is the iris diaphragm. Make sure you understand that the iris, what the iris diaphragm does, it regulates the amount of light that is reaching the specimen. All right, and then we have some sort of light or lamp source. So some sort of light source. So it could be a light, a lamp. It could be an LED light. It could be a mirror, like in olden days. And I actually have, I think, a couple microscopes that still have that use mirrors. So they have to use the ambient light in the environment to like. And so like if you wanted to use a candle or just the window light would work for that. Let me see what else. Oh, this here, this is called the base. So it's one that I want you to add to your list of parts. It's got a lot of the electronics for the cord, like the cords and the lights there. It's remember also going to be the, if this is our microscope, that we're going to put our one hand on the arm, one hand on the base, and that's how we're going to always hold our microscope so we can protect it. And while I'm here, cheers. All right. So I think we got all those parts. So that takes care of that section. So pause it if there's anything you need to fill in. And the next thing we're going to talk about is magnification versus resolution. So I think everybody kind of understands these terms, but magnification is referring to how many times you make something enlarge, how many times you basically blow it up so you can see it. And remember for magnification that it's going to be your objective lens power times your eyepiece power, or I should say, this is, remember, we're going to call it's not that eyepiece is wrong, but we want to use the term ocular times the ocular is going to give you the total power of the total magnification. So if we do these two problems here, and so if we, this one's easy, I can do this one in my head. I should be able to do, oh, actually, I know now. Okay, so 60 times 10 gives us 600 times magnification is the total magnification here. And 40 times 15 is also 600 times magnification. So this is two ways about getting about the same magnification. And it depends on the objective lens times the eyepiece or ocular lens. All right, so that's magnification. Pretty straightforward. You have to multiply the powers of those two lenses. Resolution is how clear you can see details as being separate from each other. So imagine that these two things are looked at with some sort of lens, whether it's a camera lens or a microscope lens. And in one set of lenses, you see this fuzzy figure, and it's hard to tell that those are two distinct things. It's kind of um, like headlights in a distance might seem like they're just one light, but when you get closer and they get closer to you, then you can see they're distinct and separate lights. All right. So, in this lens that took this picture, it's not obviously not actually a picture, but imagine it was that we have high resolution. We can see these as being two distinct structures. They're very clearly separate from each other. Where low resolution is, in this case, even though it would be the same object, it's in low resolution. We cannot distinguish them as being separate. And it says, factors like dirty lenses can reduce resolution, as you can imagine. But mostly, resolution is going to be a function of a particular lens. So, like, the more expensive a lens, usually the better resolution it can have. All right. I'm trying to decide what I want to do for this. You know what? I am going to skip calculating magnification. If we do this, I am going to want to do this in the actual in person. You can skip questions 3, 4, and 5 for this point. Um, but it does show you how to do it. So if you're interested, I can certainly show you the answers. I just don't think I'm going to get much bang for my buck in explaining that. It's just kind of going through like how you would calculate that. All right. So, but I actually do like question 6 and 7. So, all right. So the so if we think about it, if let's say we have two cells here, and they are um, we can see them under a lower power as being clearly separate cells. And then we go and we magnify it, and it's really like blurry. Imagine same cell, and we can't really tell them as being two separate cells. So it doesn't necessarily help us to see it bigger if it's not going to be some degree of also being clear. So it's, if you wanted to see something internally here, then that might be useful because you're enlarging it. But if it's to the point where it's so blurry that you can't see it, then that doesn't make a lot of sense. So that's kind of helps you with number 6. I'm not going to write it all out because it's just a bunch of scribble. It's going to look like a bunch of scribble. And then let me see here. So, and you need to make sure when you are going through the powers on the microscope that you start with the lowest and you go to the highest, because if you don't see anything under the lowest power, if you don't see something clearly, it's only going to get worse in terms of resolution when you're more magnified. So you really have to make sure it's very clear when you start at the I'm going to write total magnification when you start at 40, and then you're going to go to 100 times magnification. And I actually sometimes just start at 100. That's like a good starting point, but most books and in labs will tell you to start at 40 and then go to 100 and then go to the blue, which is the highest power. And remember, we never, ever, ever use our coarse adjustment, our big adjustment knob, when we are in under the highest power. All right. So it's really important. Like I said, it's only going to get worse in terms of how clear something is as you go through. So if you don't see it under 40, you're not going to see it any better under 400 for the things that we're looking at in this class. All right. So let me see what I got on the next slide. And I think that was it for this. So go ahead and fill that in. And then you're going to go on to the Pivot Interactives and do that work that is listed there. So I hope this was helpful and have a good day.